In this video, we will be taking a deep dive into the structure and function of human phosphoglucomutase isoform 1, also referred to as PGM1. Phosphoglucomutase has a molecular weight of 61,449 and a theoretical pi of 6.30. Phosphoglucomutase falls into the isomerase class of enzymes, as it interconverts glucose 1-phosphate and glucose 6-phosphate via a glucose 1,6-diphosphate intermediate. This is especially important in glycogenesis, as glucose 6-phosphate must be converted to glucose 1-phosphate for the synthesis of glycogen. Conversely, glucose 1-phosphate is converted to glucose 6-phosphate to enter the metabolic mainstream and be used as fuel in processes such as glycolysis. The reversibility of this reaction makes the enzyme vital to both glycolysis and gluconeogenesis. It is also important to note that the catalytic activity of phosphoglucomutase is reliant on a divalent metal ion cofactor, most commonly magnesium. This metal ion assists in generating a nucleophile from the phosphorylated serine, shown in orange on the left, which we will observe later to characterize the active site. Therefore, PGM1 operates via metal ion catalysis. We will take a closer look at each level of protein structure in order to learn more about how the structure facilitates PGM1's functioning. Let's start with primary structure, as Dr. Anfinson's 1972 ribonuclease experiment proved that all of the necessary information for determining tertiary structure is encoded in the primary sequence. Using the annotation filters on UniPro's protein data bank, we can identify all of the hydrophilic, polar, and/or charged amino acids in the primary sequence, as seen highlighted in pink. These will prefer interactions with the solution and base the exterior, while the hydrophobic residues, now highlighted in blue, will bury inside the interior of the protein due to hydrophobic interactions. This organization, based on hydrophobicity, is thermodynamically favorable and decreases entropy of the system defined as solution and enzyme. The active site, highlighted in red as a single serine residue at position 116, importantly is also marked as a modified residue, now highlighted in blue. This is referring to the phosphoryl group which modifies the serine and is donated to the substrate to create the glucose 1,6-diphosphate intermediate. This phosphorylated serine residue is located to the left of histidine and asparagine residues, which are both charged, creating a sequence of 3 polar and consequently hydrophilic amino acids. Short sequences of hydrophobic residues, highlighted in blue, lie to the right of asparagine and to the left of the active site serine, and help to contextualize the location of the active site even further. While the hydrophobic residues on either side bury the active site inside PGM1, the charged hydrophilic residues encourage interactions with the solution and facilitate binding with substrate. Applying UniPro's metal binding filter on the primary sequence reveals that the stabilization of the magnesium ion in the metalloenzyme is also reliant on 3 aspartate residues highlighted in pink, in addition to the phosphorylated serine. The negative charges of these acidic residues can be visualized in tertiary structure later to have ionic interactions with magnesium to stabilize it in place. By using the filter to identify the helical and beta strand sequences, highlighted in pink and orange respectively, we can also predict the secondary structures. A quick glance at the annotated primary sequence suggests that there are many more alpha helices contributing to the secondary structure than beta sheets. Let's take a closer look at the secondary structure to see how this manifests physicochemically. Phosphoglucomutase is comprised of alpha helices, anti-parallel beta sheets, and parallel beta sheets. These secondary structures can be observed to be connected by loops and turns. Each of the green frames highlights these secondary structures. In the top left frame, there are 2 full alpha helices. In the top right frame, there's a parallel beta sheet denoted by 2 gray arrows pointing in the same direction. And in the bottom left corner, there is an anti-parallel beta sheet denoted by 2 gray arrows pointing in opposite directions. The bottom left frame also highlights a loop connecting the secondary structures. Now, without jumping too far ahead, I believe now is a good time to note that loops play an important role in PGM1's active site. The loop on the leftmost side of the frame contains the phosphorylated serine which binds to the substrate, whether that be at carbon 1 or 6, and receives the lost phosphoryl group from the diphosphate intermediate to return to its initial state. Additionally, The loops above, below, and to the right of magnesium in this frame are responsible for positioning the 3 aspartate residues as highlighted in the primary structure for stabilizing magnesium. Now let's take a step back and look at the entire enzyme to get a better understanding of the tertiary structure. The tertiary structure of PGM1 consists of 4 domains, relatively similar in size, with the active site located in the cleft where the 4 domains meet. The hydrophobic interior protects the active site from water molecules, which could hydrolyze phosphoester bonds formed during enzymatic activity. The overall 3-dimensional conformation of the protein resembles a heart shape. This is why PGM1 is my favorite enzyme. We can see that the tertiary structure is dense with alpha helices, especially at the bottom of this frame in the blue and red domains, creating the pointed base of the heart shape. The tighter helical structures makes approaching the active site from underneath nearly impossible. The green and yellow domains are characterized more so by beta sheets and open up the heart-shaped enzyme's cleft to the exterior solution and help to shuttle substrate into the active site. Hydrogen bonds between R groups, such as the 2 formed between aspartate and threonine, can also be visualized to be importantly contributing to tertiary structure. Phosphoglucomutase lacks quaternary structure as PGM1 functions as a monomeric protein and does not interact with any other polypeptide chains. In summary, phosphoglucomutase is an enzyme essential to the interconversion of glucose 1-phosphate and glucose 6-phosphate via a glucose 1,6-diphosphate intermediate. It is essential to glycolysis and gluconeogenesis and operates via metal ion catalysis as it relies on a divalent metal ion cofactor such as magnesium for its catalytic activity. The active site is characterized by a post-translationally phosphorylated serine, which interacts directly with substrate to donate its phosphoryl group to create the diphosphate intermediate. Loop structure motifs stabilize metal binding of the magnesium ion cofactor necessary for enzymatic activity. The hydrophobic effect results in the hydrophilic amino acids on the exterior and a hydrophobic cleft, which protects the active site from water molecules which could hydrolyze phosphoester bonds formed during enzymatic activity. PGM1 is a monomeric protein that lacks any other interactions with polypeptide chains to constitute a quaternary structure. The 4 domains which comprise PGM1's tertiary structure take the shape of a heart when folded properly. This is why PGM1 is my favorite enzyme.